Welcome back, everyone. So we're talking about gene regulation. We just talked about the lactose operon, the lac operon, and that is a series of enzymes used to digest or break down lactose. So we only need to catabolize lactose when it's present. That's very different from what we're looking in biosynthetic pathways. So something like tryptophan, an amino acid that we're not trying to break down for energy. We are trying to build it up to be able to make proteins for ourselves. It's going to look a little bit differently how we handle these operons. So operons that code for enzymes involved in catabolic pathways, like the lac operon, are going to look pretty similar to being controlled that same way. They'll be inducible, only turned on when that energy source is present. Operons that regulate enzymes involved in anabolic pathways are generally repressible operons. So the default state is we need to make these components for our cell, and only when we have plenty of it built up does our cell respond by saying, okay, I have enough, I don't need to keep making it. And that end pathway, that end product of the pathway will repress that operon. So these are repressible operons. The tryptophan operon includes genes involved in creating the enzymes to make tryptophan, and also some regulatory sequences, the operator and promoter. And the effector molecule here is the end product of the pathway. It's tryptophan itself. So when we are having plenty of tryptophan in the cell, tryptophan itself is able to repress the pathway. And when tryptophan is not present, the pathway is turned on, and we're going to make these enzymes to make tryptophan. So without tryptophan, we make tryptophan. If you've got plenty already, you stop making more. The regulatory gene for the Trp operon is called Trp-R, and this is a little bit different. Instead of binding without the repressor, it's actually activated or binds to the operator DNA when it's bound to tryptophan. So it has to be bound to the effector, and that's when it will bind to the DNA and prevent transcription of the operon. So it works a little bit differently from how our lac operon did. So tryptophan here is considered a co-repressor because you need not only the repressor protein but also tryptophan to shut off the gene expression for this operon. If you have the repressor without tryptophan, it doesn't bind to DNA, and so the RNA polymerase will bind to the DNA and allow transcription of these genes. So you're now making the enzymes necessary to make tryptophan for your cell. So the full-length tryptophan mRNA is made when tryptophan is scarce or absent. So the enzymes in the tryptophan biosynthetic pathway are made so that you can make tryptophan. You don't have enough, you're going to make more. When tryptophan is present, the production of the tryptophan mRNA is inhibited. However, the leader sequence is produced even when tryptophan is present and the Trp operon is inhibited. The presence of a control element within that leader sequence that determines whether transcription will continue is called attenuation. So the control element in the leader sequence is more sensitive than the repressor mechanism in determining whether there's enough of the effector molecule, enough of that tryptophan present to support protein synthesis. So this is a pretty common type of control for amino acid biosynthesis. So a portion of the leader sequence will be translated, and you'll form a leader peptide that's about 14 amino acids long. And the leader sequence for this mRNA contains 2 adjacent codons for tryptophan, so you'll put 2 tryptophans in it. And it also has 4 regions, numbered 1, 2, 3, and 4, that will pair up to form hairpin loops. Regions 3 and 4 of the leader mRNA plus 8 more adjacent U's will make a terminator, and they can create a hairpin loop that acts as a transcription termination signal. When tryptophan levels are low, the ribosome will start translating this polypeptide, and when it gets to those tryptophan codons, it'll pause, waiting for that tryptophan to be incorporated. But because the levels are low, it's going to be waiting a longer time. So that stalling is going to block region 1 and promoting the hairpin to form between regions 2 and 3. That is the anti-terminator hairpin. When region 3 is paired with 2, it can't form the termination structure with region 4, and so transcription by RNA polymerase is going to continue. So that is one way that when tryptophan is absent, the transcription and translation of that gene continue. So that results in making the Trp Operon mRNA transcript. But if tryptophan is plentiful, that tryptophan gets put right into the ribosome, incorporated into the growing polypeptide, and instead you get the hairpin forming with regions 3 and 4. That terminates transcription. So you've attenuated that operon because you already have plenty of tryptophan available. Sigma factors are also used to determine which sets of genes can be expressed. You'll remember talking about sigma factors when we talked about promoters for our bacterial cells. So they have different sigma factors to control which genes are going to be transcribed. The sigma factors recognize those different gene promoters. The most common one in E. coli is sigma 70. And there are alternative sigma factors that alter promoter recognition that let your cells adapt to different conditions. So if the temperature is warmer or cooler, there's more UV radiation. Their environment is changing pH. They're in nitrogen limiting conditions. They have to grow slightly differently to be able to survive. They can have those alternative sigma factors to activate those genes that allow them to survive in those changing conditions. Different types of bacteria can have different numbers of alternative sigma factors. And some can even be produced by bacteriophages, those viruses of bacteria that can allow a phage to take over the transcriptional machinery of the cell. Okay, so now we've explored some operons. We're going to explore a little bit more of some gene control in our prokaryotes and then jump into eukaryotic cell conditions to explore how we control genes in our own body. I'll see you later. Bye.